and thank you for coming. Hello everyone, my name is Yu Ito. I'm from Osaka, Japan. And well, I have no copyright, so you feel free to take photos. Once you take photos, send me later. I'm here alone, so. And today's topic is about Monochoria. I've been working for aquatic plants since I was a student, and it's been 10 years, 15 years, but I'm talking about this Monochoria species, which is mostly Asian species, so I'm pretty sure you are not familiar with this. Well, here's the outline of today's talk. I'm going to give a brief background, and then move on to the Monochoria species introduction, and then move on to previous studies and the aim of the study, and then materials and method, and results of discussion. I have 1, 2, 3 results of discussion, and the future direction and acknowledgement. Right. So here's the background of my study, why we work, how we work, and where we work. What we want to know is to understand the biodiversity in the world. So let's say I want to know the exact number of species in the world, or let's say the exact number of aquatic plant species in the world. And here you see a website from the Kew Botanic Garden. This one says that every year we have 2,000 new species. So I'm pretty sure that our study is among these 2,000 new species. So how we work? I'm a taxonomist, I'm a systematist, so I'm working on with molecular phylogenetic study, sometimes in collaboration with the ecological data, morphological data, and sometimes probability data. But today's much focus on molecular phylogeny plus ecological data. And where we work, I was born in Japan, so I focus on the Asians. And you see the biodiversity hotspot here. We have a Japan biodiversity hotspot on the Philippines, and the Malaysia, and the Sunda, the Indo-Burma, and sometimes in the Western Ghats and Sri Lanka. These are the area of focus in this study. So, you see the Monochoria species, the beautiful flowers. So probably you can see it in Europe, in like Botanic Garden or some places. But basically, the distribution patterns, as you see, mostly Asian distribution, plus Australian and some 1 or 2 species in Africa. You see the plant is very beautiful flowers, so it's easy to find. And mostly in Asia, the plant can be found in rice field or pot field. So, and if you go like a dry season, you don't need to get it. You don't need any specific gear. You can just grab hand. But sometimes, like in rainy season in tropical Asia, you need to go swimming. Like this one, I do remember when we found this in Laos. It's deep, so I got wet when we collected it. So now I remember that in the lecture, we needed to remember to get wet. So I did it. But for others, it's not difficult to practice. And according to the monographic work, I think it's 20 years ago or something like that, and 8 species are recognized in the world. Only 8 species. But we'll see, we do have more species, I think. So here is the previous study. It's a very important study. A few years ago, a Chinese researcher published this paper. It looks good. And they collected many specimens, and one from Australia, unidentified specimen, and then some Pontederiaceae and Africa specimen included, and the Asian species are included. And you see the molecular phylogeny based on chloroplast and sequence data. So it looks good, But I do have some doubt that if or whether this taxon sampling really cover all the diversity of this genus. The other thing is they use only chloroplast DNA sequence data. We do have nuclear DNA sequence data. So let's see if the chloroplast phylogeny reflect the species phylogeny or not. And here's the flora of China treatment. Based on the morphological data, they said 4 species. But we'll see. So this is the aim of this study. What we want to know, the number of species of Monochoria revisited. I mean, the Chinese researcher already did it. So the second one is area of species diversity in Monochoria, with which part of Asia the diversity is in. And materials and method. We did it from Japan to Myanmar to India and Indonesia and Australian specimen included in our taxon sampling. And based on using the morphographic work, based on morphological data, we can identify the specimen into 5 species, 8 out of 5 species. But we do have multiple samples for like a widespread species, Monochoria vaginalis from Japan to Australia and Monochoria hastata. We collected widely from the tropical Asian countries. And we use this taxon sampling for the subsequent analysis. And for method, we did the molecular phylogenetic analysis. And here you see the chloroplast DNA phylogeneity visited. And then the our taxon sampling plus Chinese researchers' sequence data is included. So we do have Australian specimen Monochoria as a base. And then interesting one is here, it's Laos unidentified specimen, which is very unique genetically. So I'm pretty sure this one is the subject for new species. And here we have Australian unidentified specimen, and then this one of these is from the Chinese researchers' data. And we have Monochoria Pontederiaceae species. Here it is. This one is interesting because this name used to be a synonym of another widespread species, vaginalis. But I'm pretty sure this one is different from this one, vaginalis. And then a Chinese researchers' sample is among these. And here we have a larger species. This one, one is our sampling, and then the others are from the Chinese researchers. The most interesting one is this one. We call this Monochoria hastata. No specimen from Chinese researchers' analysis. The thing is, but they do have a hastata. Their hastata is here, together with what we call Monochoria pseudo-hastata. I'm pretty sure the real hastata belong to these, but they said the hastata belong to this. I'm pretty sure this one could be the second case of new species. And here we have Monochoria, for example, from Japan, and Monochoria vaginalis from Japan to Myanmar. So let's say it used to be 8 species, but probably the species number could be different from the monographic work. So we test using another data from nuclear DNA sequence data. You see the top 6 nuclear sequencing and the 5' A and 5' C, and not really cancer sampling, but we see like the top 6 phylogeny, the species as we found in the chloroplast DNA phylogeny is mostly supported by this top 6 nuclear sequence data. So we use the chloroplast DNA sequence data plus this nuclear top 6 data to see the species delimitation analysis. Now here you see the heat map. This analysis gives me the clue that the which specimen belong to which species. So now you see here, this specimen may belong to the one species, and this one must be unique, this one must be unique, this one must be unique, and this must be clustered into one species. So mostly, as I show you in the chloroplast DNA phylogeny, we do have 12 sampling, 12 species in the world. So here is the revised taxonomy of the species, and we have morphologically 8 species, but now we do have 12 species, and an uncollected one, Australian species, and Australian African species. But except the 3, we do have 9 species in the world, in Asia. The most interesting one is the, as I mentioned, the Laos specimen. It's not identified so far. And then the other one is what we call the pseudo-hastata. This must be a new species in the world. And here you see this area, like Laos to Vietnam, have many different species. Compared with like Japan, there's only 2 species in Japan. So now you see the ecological data analysis. We applied ecological niche modeling to see the 2 similar species. We do have 2 cases. Here it is the hastata and pseudo-hastata used to be the same, but we do have a different evidence to separate these into 2 species, basically, based on genetic data. And you see the different, slightly different distribution pattern. And these 2 more clear differences. Here is the Pontederiaceae, and here is the vaginalis. It used to be the synonym of vaginalis, but now we have a deep evidence that these are different species. And here, the vagina is mostly distributed in Japan, but in the other hand, the Plantaginia mostly distributed in tropical Asia. So, what we want to know in the future is to see the micro biodiversity hotspot or nano biodiversity hotspot. Now we have this kind of wide range of, a wide, like Indo-Burma biodiversity hotspot, but we want to know exactly where the nano biodiversity spot is, for instance, in the Indo-Burma biodiversity hotspot. We can apply this analysis in the future. Well, here's the acknowledgement, and thank you for listening. If you have any questions, don't hesitate to ask me. Thank you.
Yeah, we have time for questions.
As a taxonomist, I don't have a question. Do you plan some morphological study? Because when you know from the DNA that this is the one clade, this is the other clade, so usually it's possible, or often it's possible to find some minor differences which are not known before or were overlooked because people didn't know that this is one and this is second. So do you plan something like that to search for differences?
I have a confidence to give a revised morphological key to the species, except the Laos specimen. We have only one specimen, so we are very hard to find the morphological variation of that species. But in the case of like Shuddha hastata, what we call, we do have a difference between hastata and the Shuddha hastata morphologically. So I'm pretty sure I can use morphology to do the taxonomy. Thank you.
Other questions?
What do you think of the recent placement of Monochoria into Pontederiaceae and Sulato?
Yeah, that's sorry.
Sorry, it's
No, it's okay. Let me explain. I started this research 10 years ago.
Okay.
At that time, from this, we continue with calling Monochoria. So I have to say, I have to follow the paper saying that's the Pontederiaceae since Sulato. So if I give the taxonomic study, probably I have to say the Pontederiaceae, Shuddha hastata or something. Yeah, that's if we follow that paper or not. So, well, thank you so much.
Was your heat map, was that Stacy, the R package?
Yes, Stacy. Yes.
And was that multi-locus or single locus?
Yeah, multi-locus.
Okay, nice.
We use the chloroplast DNA plus topo 6 nuclear DNA.
So you combine the chloroplast and the nuclear together?
It's not combined.
Okay.
As far as I remember, it's parallelly analyzed with different locus data to infer the species phylogeny as well as hidden one.
Okay.
So yeah.
It was nice result. It was nice.
Thank you.
One more, if anyone has any.
And no sign of hybridization in these genes?
Yeah, as far as I know, I mean, I see the chloroplast phylogeny, I see the nuclear phylogeny. There's no differences between the two. And then, on the other hand, as far as we think it's nuclear phylogeny, there's no kind of double peaks happening. So probably there's no so far.
Chloroplast is maternal, so there cannot be hybridization. It's just from mother.
No, I mean, we do have nuclear phylogeny. As far as I know, if something happened, like the nuclear phylogeny, this species belongs to here, but the chloroplast DNA is from here, so that we get something happen. But we didn't find such kind of evidence, so I guess there's no hybridization.
No sign. Okay.